Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So, in this catabolism of the carbon skeletons of amino acids lecture series, so in this particular lecture, I will be going over amino acids that produce fumarate. Let's get into the details. So, amino acids that produce a fumarate as one of the TCA cycle intermediate is phenylalanine and tyrosine catabolism. Already we have seen this catabolism of phenylalanine and tyrosine in my previous lecture, where I was explaining the amino acids catabolism that will give ketone bodies, acetoacetyl-CoA or acetoacetate as a ketone body. So in that particular lecture, I have already explained you how exactly phenylalanine and tyrosine will go into catabolism. So let me recap that in this lecture, because both phenylalanine and tyrosine will produce fumarate along with acetoacetyl-CoA or acetoacetate, which will go into ketone body. So now let's look into the details of reactions. So the phenylalanine hydroxylase enzyme, it converts phenylalanine into tyrosine, as it is shown here in the reaction. Phenylalanine converted to tyrosine by an enzyme phenylalanine hydroxylase, and this phenylalanine hydroxylase is a monooxygenase system of enzyme where an atom of oxygen is incorporated into a benzene ring to introduce a hydroxyl group here in the benzene ring. So the difference between phenylalanine and tyrosine, as you are aware, is only the hydroxyl group. Now, in order to do this, a phenylalanine hydroxylase enzyme, it needs a coenzyme called tetrahydrobiopterin, which is shown here. I am highlighting that. And this tetrahydrobiopterin, it gives 2 protons to the reaction where an atom of oxygen is incorporated into a benzene ring to introduce hydroxyl group to tyrosine, and another atom of oxygen is incorporated with 2 protons that are coming from tetrahydrobiopterin to release it as water molecule. That's how H2O goes out of the reaction. The H2 there actually came from BH4. That's why BH4 will be converted to BH2 in the reaction. And the BH2 will have to be converted back into BH4. That will be done by dihydropteridine reductase enzyme. And the dihydropteridine reductase enzyme, it is going to reduce BH2 into BH4. For that, we need a reducing molecule. And that reducing molecule is NADH plus H plus, which will be oxidized into NADP plus. So this is how a sufficient BH4 will be maintained in the cell so that phenylalanine hydroxylase continue to convert phenylalanine into tyrosine. And also note that, like any other coenzymes in our cells, there will be limited amount of coenzyme being synthesized in the cells. That means coenzymes need to be recycled all the time. So the same concept applies to tetrahydrobiopterin also. So a new molecule of tetrahydrobiopterin can be synthesized by a multiple reaction that will go from guanosine triphosphate, GTP. GTP can undergo a reaction, multiple reactions to give tetrahydrobiopterin. That's how tetrahydrobiopterin is synthesized, and later it will be recycled using dihydropteridine reductase enzyme. Alright, so these are some of the reactions in the conversion of phenylalanine into tyrosine that you must remember. There are a lot of clinical applications to this particular reaction here. So let me explain you briefly. Let me introduce the clinical applications. What are the clinical disorders that can apply in this conversion of phenylalanine to tyrosine? So the enzyme phenylalanine hydroxylase, if it is absence first I will write absence of phenylalanine hydroxylase, I will write it as PAH. Phenylalanine hydroxylase. Absence of phenylalanine hydroxylase will give rise to type 1 phenylketonuria, type 1 PKU. Okay, now let me rewrite in a different way so that it will be easy for you to recall things. So, phenylketonuria is a disorder associated with the conversion of phenylalanine into tyrosine. Phenylketonuria. We are going to go over this disease in detail later. So, there are 4 types of phenylketonuria. Type 1 phenylketonuria, type 2 phenylketonuria, type 3 phenylketonuria, and type 4 phenylketonuria. These are 4 types of phenylketonuria. What are all the causes for each one of them? So, type 1 phenylketonuria is because of absence of PAH, phenylalanine hydroxylase enzyme, absence of PAH. Type 2 phenylketonuria is because of deficiency of PAH. Type 3 phenylketonuria is because of deficiency of dihydropteridine reductase. I will write it as DHPR, dihydropteridine reductase. That's this enzyme here. And type 4 phenylketonuria is deficiency in the secretion, in the synthesis of BH4. Deficiency in the BH4 due to decreased conversion of GTP to BH4. Okay, so that's what, this is how we can divide a phenylketonuria into 4 different types. Okay, so let me write down here in the pathway where exactly all this type 1, 2, 3 and 4 will come. Type 1 and 2 will be here. One is absent, other is deficient of phenylalanine hydroxylase. So, phenylalanine hydroxylase, if it is absent, it is type 1 phenylketonuria. If it is deficient, it is type 2 phenylketonuria. The type 3 is because of decreased activity of dihydropteridine reductase, DHPR. DHPR is here, PAH is here. Okay, and type 4 phenylketonuria is here, conversion of GTP into tetrahydrobiopterin. So, this is how a phenylalanine conversion into tyrosine may lead to certain types of phenylketonurias. We are going to go into details later. Alright, so once we get tyrosine, so what will happen to the tyrosine? So the tyrosine will undergo further catabolism to make a fumarate and acetoacetate. Okay, so that's shown in the slide here. Phenylalanine is converted into tyrosine, and tyrosine undergoes tyrosine transamination reaction to become p-hydroxyphenylpyruvate, and that p-hydroxyphenylpyruvate is converted to 𝐒𝐲𝐿 by p-hydroxyphenylpyruvate dioxygenase enzyme. 𝐒𝐲𝐿 is converted into methyl acetoacetate, and methyl acetoacetate is converted into fumaryl acetoacetate, and fumaryl acetoacetate is converted to fumarate and acetoacetate, and acetoacetate is a ketone body, fumarate is a glucogenic. That's where fumarate comes in here. So the purpose of this lecture is to show how exactly phenylalanine and tyrosine is converted to TCA cycle intermediate fumarate. So fumarate is, it can go into glucose formation later, and acetoacetate is a ketone body. Alright, so while we are looking at these conversion of phenylalanine into tyrosine, I have already told you there is a risk of the disease called phenylketonuria. Conversion of tyrosine into p-hydroxyphenylpyruvate done by tyrosine transaminase enzyme will give rise to tyrosinemia type 2. And then conversion of p-hydroxyphenylpyruvate into 𝐒𝐲𝐿 by p-hydroxyphenylpyruvate dioxygenase will give rise to a transient tyrosinemia, that is type 3 tyrosinemia. And then homogentisate into malonyl acetoacetate. Enzyme is homogentisate oxidase. Deficiency of that enzyme will give rise to alkaptonuria. Malonyl acetoacetate converted to fumaryl acetate by isomerase reaction. And then fumaryl acetate to fumarate, it will be done by fumaryl acetoacetate hydrolase enzyme, and that will give rise to type 1 tyrosinemia, which is also called as tyrosinosis. So there are a couple of disorders here that we are going to go over later. That is phenylketonuria, that's number 1. Then number 2, the important ones is type 1 tyrosinemia called tyrosinosis. Number 3, alkaptonuria. Okay, so this is how a tyrosine is converted into fumarate, which is glucogenic, and acetoacetate is a ketogenic. So that's how amino acids, that is phenylalanine and tyrosine, will make fumarate as the TCA cycle intermediate. That's about it. Thank you.